Hello. Thank you, Professor Ji-Hyun Kim, for the kind introduction. I also thank the organizers for inviting me to give a talk in this wonderful conference. My name is Keehyoung Joo, and I work at the Korea Institute for Advanced Study. My research focuses on using AI and computational methods to study proteins. The title of my talk is DeepFold PLM. I add PLM. Yeah. This subtitle is Recent Advances in Protein Structure Prediction and Beyond. I'd like to begin with this quote. It is God's privilege to conceal things, but the king's pride is to research them. And this reminds us that what is research or science? It is like finding hidden treasures in nature, and we can share king's pride with joy. First, as we all know, this year's Nobel Prize in Chemistry was awarded to David Baker, Demis Hassabis, and John Jumper for their groundbreaking work on proteins. David Baker focuses on designing proteins, while Hassabis and Jumper developed an AI called AlphaFold to predict protein structures. In this talk, I'll focus on protein structure prediction and explain our contributions to this field. Let's go back to where this all started, from AlphaGo. In 2016, AlphaGo defeated a human professional Korean Go player, Lee Sedol, showing the power of AI. In 2017, Hassabis said, while it is still early days, AlphaGo Zero constitutes a critical step towards this goal. If similar techniques can be applied to other structured problems, such as protein folding. He specifically mentioned about protein folding as a problem that AI could help solve. I also agreed with his opinion, but I never expected that he would solve this problem so quickly with his DeepMind team. Fast forward to 2020, they achieved this with AlphaFold 2 as a solution to a 50-year-old grand challenge in biology. AlphaFold 2 represents a breakthrough in protein structure prediction, achieving near experimental accuracy with GDTTS scores approaching 90. This animation shows superpositions between experimental structure in green and computational predictions in blue. We can see they have nearly identical shapes. It is content of my talk. Looking at today's audience, I noticed how diverse it is. So I thought a brief introduction to protein structure prediction might be helpful. In these pictures, let me explain protein folding. It is like paper folding called origami, but with big difference. You can fold a paper like this into infinite number of shapes, like a bird, a plane, boat, and etc. But proteins fold into just one unique shape for given their sequences like this. I will explain more with the history. We are familiar with the central dogma of molecular biology, which represents the logic of biology. It was proposed in 1958 by Francis Crick, who discovered the structure of the DNA double helix with his colleague Watson. Proteins are synthesized in the cell. DNA encodes RNA, which translates into proteins. However, while DNA provides only the sequence information of protein, it does not specify a 3D structure of protein. The first protein structures to be resolved were myoglobin and hemoglobin, achieved by Max Perutz and John Kendrew. They used X-ray crystallography to determine their 3D structures. Over the past 50 years, experimental techniques like this have continued to unveil the structures of proteins, contributing significantly to our understanding of biology. As a result, an incredible amount of protein sequences and structural data has been accumulated. So, we have another central dogma: sequence, structure, and function. Today, we have in UNIREP 100 database contains over 365 million protein sequences. However, only about 227,000 3D structures have been deposited in the PDB database. This large gap highlights a significant scientific bottleneck. Christian, Christian Amundsen proved that a protein's shape is determined solely by its amino acid sequence. It means that protein structure is its free energy minimum state in biological environment. The Levinthal paradox is another challenge. How proteins fold so quickly into a single structure, despite countless possible conformations. This would be impossible if all possible conformations were explored. Proteins are vital for catalysis, switching, binding, and structural support, making their structure key to understanding their functions. It's truly a protein world, the molecular machinery of life. This working protein is the kinesin motor protein, which acts as a truck, transporting cellular cargo within the cells. The highway it moves on is also made of proteins. Here is the human MHC protein, a key player in the immune system. It called as a collagen, which is essential for forming muscles in our body. Its triple helix bundle structure makes it incredibly strong and hard to break. The spike protein is quite famous during the COVID-19. It sits on the coronavirus surface, enabling it to invade respiratory cells in humans and animals. To emphasize once more, a protein's shape determines its function and how they work. Experimental methods like X-ray crystallography, NMR spectroscopy, and cryo-EM have been used to determine protein structures, leading to many discoveries. We have also worked to predict protein structures using two computational approaches, knowledge-based and physics-based methods. Template-based modeling rely on the idea that similar sequences often have similar structures. This case, where even low sequence similarity with just 15% identical residues can result in high structural similarity. AlphaFold also builds on this principle. If the sequences are similar, their structures are also similar. Quite very simple. Yeah, this is the AlphaFold 2 paper published in Nature. John Jumper and Demis Hassabis here. AlphaFold 2 is an end-to-end model that predicts protein 3D structures from input sequences, while the input starts as a single sequence. The model also uses two key types of data: multiple sequence alignments, we call the MSA, and template structures. Among these MSA, alignments of similar sequences are the most critical input. The network architecture includes the evoformer, an attention mechanism that processes the input data to generate two types of embeddings, single representations and pair representations. These embeddings are then passed to the structure module, which predict the 3D structure of protein. Let's take a closer look at the critical role of MSA. Finding similar sequences from a database for query sequence. Sequence is a standard query sequence is a standard method in bioinformatics. MSAs are essential because they reveal patterns related to protein 3D structure. For example, when two amino acids I and J are close in 3D space, their mutations in the column often correlate in the MSA. It is called correlated mutation or coevolution. This concept forms the basis of physical models like the patch model. Is an extension of Isaac model. The Hamiltonian energy function describes interactions. Hj represents self potential and Jij represent interaction potential linked to distance distributions. Now, these are visualized in a histogram where bright green areas indicate two amino acids are close in 3D space. AlphaFold 1 used this framework with a CNN-based network for histogram prediction. AlphaFold 2 advanced this by using an attention mechanism to model interactions, leading to remarkable accuracy in protein structure prediction. Here's the diagram of DeepFold we developed 2 years ago. I'll only give a brief summary here. DeepFold improves protein structure prediction by modifying loss functions and template alignment method based on our data analysis. Additionally, we refine protein structures further through energy function optimization. For more details, please refer to our published paper. This slide shows a successful example of DeepFold prediction. This is a visualization of MSA for this target. The x-axis represents the sequence positions, and the y-axis represents the sequences in the MSA. The colors indicate the sequence identity relative to the query sequence. In this case, the quality of the MSA is very high, with an effective diversity, NEFF, of 9, and 1,000 more sequences are aligned. NEFF measures the diversity within the MSA, effective number of sequences. Simply, it indicates how informative the MSA is. A higher score means it is more informative. As a result, the predicted structure in green is highly similar to the native structures in cyan, achieving a TM score of 0.97. It means 97% correct. This score indicates an almost perfect match between the predicted and the experimental structures. This example demonstrates a case where no MSA, the standard method failed to find homologous sequences in the database. As you can see, the predicted structure captures the overall presence of helices, but the structural similarity to the native is very low. With a TM score of just 0.25, this is considered a failed prediction. To achieve successful structure prediction, the MSA must be of high quality, with an NFF score of at least 3. We need alternative methods to generate high quality MSAs in the case of this example. So we have recently developed a new advanced framework called DeepFold PLM. First, we developed a new method called PLM MSA based on protein language models to generate MSAs efficiently. I will explain this in more detail. on the next slide. Next, we re-implemented DeepFold using PyTorch with multi-GPU support to handle large proteins. We also added a protocol for predicting multimer structure. The PLM MSA algorithm works as follows. First, we use protein language models like ESM1B. It is developed by Facebook, and ENC to embed protein sequences. Next, we apply pooling and contrastive learning to map these embeddings into the vector space effectively. Then we calculate the similarity score to identify similar sequences. Finally, we use the PLM align algorithm based on the Needleman-Bunz method to align sequences with a similarity score greater than 8.0, building the MSA. We have built an embedding database using about 65 million sequences from the UNIREP 50 database, enabling this process to be efficient and scalable. To explain in more detail, we use a loss function similar to SIMCSE. It is commonly used in contrastive learning. In this vector space, the red dot represents the query sequence. Pink dots are positive sequences from the same MSA. And the blue dots are negative sequences from the same negative sequences from different MSAs. By optimizing this softmax-like loss function, positive sequences are brought closer together, while negative sequences are pushed farther apart. This helps us map protein sequences into a well-defined vector space. Using distance-based representation, we can cluster all points in the vector space in advance. This allows us to quickly find and group similar protein sequences when given a query sequence, making the process highly efficient. Another key advantage of PLM MSA is its ability to identify relevant sequences even for challenging targets where traditional methods fail. This is a technical slide of DeepFold re-implementation. I want to skip this the time limit. This shows the result using PLM MSA compared with the standard MSA search method, jackhammer, used in AlphaFold. Panel A, the x-axis represents sequence length and y-axis represents the MSA construction time. It compares PLM MSA with jackhammer. PLM MSA is about 47 times faster. Unlike jackhammer, which takes more time as the sequence lengths increase, PLM MSA operates in nearly constant time. As shown in panel B, completing most MSAs within 10 seconds. However, it shows similar performance to the recently published MM6 to GPU method. On average, PLM MSA has a slight advantage. Meanwhile, panel C shows that PLM MSA finds, it is the PLM MSA distribution, finds more, it finds more diverse sequences with a higher NEFF score compared to jackhammer and MM6-2 methods. About 30% of sequence overlap with jackhammer, but PLM MSA consistently retrieves more varied sequences. This shows structure prediction result, jackhammer versus PLM MSA. PLM MSA achieves comparable or slightly better TM scores than jackhammer on average. For two highlighted targets, A and B, where PLM MSA outperformed jackhammer, and one case C, where PLM MSA performed worse. For target A and B, PLM MSA achieved higher TM scores. This improvement is because Jackhammer found only 15 to 13 sequences for the MSA, while PLM MSA provided over 1,000 more homologous sequences, resulting in better structure predictions. However, in target C, even though PLM MSA found more sequences, the alignment included noisy gaps, we can see, leading to a poorer result. This case highlights the need for more advanced filtering method during the alignment process to improve the quality of MSAs. This is an interesting target with two domains where the packing between the domains. As shown in the figure, predicting the relative positions of the smaller domain, it is the smaller domain, was particularly difficult. However, using PLM MSA led to a better domain packing compared to the standard MSA. The two MSAs look similar, but PLM MSA had a slightly higher NEFF score of 9.7 compared to 9.4 for the standard MSA. This difference led to a much better TM score. We are also developing a web server for high throughput protein structure prediction using our PLM MSA and GPU optimized DeepFold. This service will offer fast and efficient predictions to support the protein structure prediction community. Recently, AlphaFold version 3 was released. AlphaFold 3 replaces the structure module with a diffusion module, reduces the need for MSAs, and can predict the structures biomolecules like ligands, ions, and nucleic acids. Diffusion models are commonly used in picture generation tasks add diversity to predictions. Very recently, DeepMind has shared the inference code and model parameters for academic use only. It's hard to keep up with companies like DeepMind. We need to learn and apply these technologies to drive progress in life sciences. We believe it's time to move beyond structure prediction and apply AI to study protein interactions. This includes protein-protein interactions, protein-ligand interactions, and even protein design. As we reflect on the theme of this conference, what's next for AI, I was reminded of this year's Nobel Prizes. Not only was the Nobel Prize in Chemistry awarded for AI work, but the Nobel Prize in Physics also went to two pioneers in AI, John Hopfield and Jeffrey Hinton. I heard there was a debate. Is AI physics? Yeah. As a physicist, I would like to share some of my thoughts. Richard Feynman said that all things are made of atoms, wiggling, jiggling. Later, John Wheeler introduced the it from bit theory, suggesting that the universe is fundamentally made of information. That means everything is information. And matter emerges from the processing of this information. In AI, we now focus on embeddings that is compact representation of information. AI learns embeddings directly from data. For example, ChatGPT starts with words or tokens, uses attention mechanisms. I have no time. Mechanisms to build embeddings and generate meaningful output from them. It's sometimes useful and powerful, like in the case of AlphaFold. I think science is moving from focusing on atom to word. Information is not just abstract. It can work in energy and physics. Finally, I came across an interesting passage in the Bible. It is from the book of John, a disciple of Jesus. It begins with, "In the beginning was the Word, And the Word was with God, and the Word was God. And in verse 3, it says, all things were made by him, and without him was not anything made that was made. It's quite interesting with this AI era. Oh, I'm sorry. Thank you for your attention.
Thank you very much for your interesting talk,
Yeah.
Dr. Joo. If you have any question from the floor, please raise your hand. We will deliver the microphone. In the meantime, I can take advantage of this.
Yeah. I'm sorry.
Yeah.
So
I have a very naive question. So if you have a TM score for 0.95 or 0.7, is there any set you want to get to? And then if you have a lower than ideal optimal TM score, what could be your feedback loop for the increase your TM score, especially specific, protein species? Because I don't know anything about these prediction model. Maybe I sound too much like just outside scientist, but please.
Yeah. TM score means actually the correctness of the protein structure between predicted structure and the native structure. Native structure means the ground truth data. Yeah. We need to improve this method to get a more higher TM score. Yeah. I think there are also more rooms to improve. For example, we need to improve the details of the protein structure. For example, side chain. Yeah. Actually, there are many details of the protein structure. So we need to research more to improve the details of the structure, to apply the other protein research. Sorry, I'm sorry. I overused your time. Yeah, we can discuss more later. Thank you. Yeah.
Let's thank Dr. Joo again. Thank you very much.